proteins that are going to trap the water and keep it from flowing. Can we have coagulation in serum? No. Because serum is taking out specifically the coagulation proteins. So serum will not coagulate, all right? Now, if we look at these in a little bit more detail, here's the vascular phase. Okay, it's just a natural reflex of the damaged cell walls of the smooth muscle. And then the platelet phase you have this diagram in your lecture notes. These are the platelets responding primarily to the irregular surface created by the cut vessel wall. So in the loose connective tissue of our tunica intima, we have collagen fibers, and they're exposed when the vessel wall is cut. In addition, platelets will stick to an irregular surface, such as an inflamed vessel like phlebitis, all right? Or we talked about atrial leading to blood clots, where the platelets tend to settle down out of the blood, and those little trabeculae carni that interfere with the clean flow of the blood. And as they do so and come in contact with the collagen, that triggers an internal reaction that releases the three major chemicals of platelets, which are serotonin, ADP, and thromboxane. Serotonin is, we're most used to hearing about serotonin as a neurotransmitter, okay? It's the feel-good neurotransmitter in the brain. But it also is made by the megakaryocytes, which, by the way, heads up, which I forgot to tell the morning class, so you're already ahead of them. On your lecture exam is a matching, set of matching, where I ask you to match cells to the molecules that they secrete. So platelets or megakaryocytes would be releasing these components. B lymphocytes would be releasing antibodies, IgE or immunoglobulins. We'll talk about neutrophils, and of course basophils make histamine. So start kind of making a list of the different molecules that are made by the cells that you're going to talk about.
You said match molecules to chemicals that they secrete?
Yeah, so they're secretory components. All right, so serotonin, as a release from the platelets, functions to increase the vascular spasm. ADP, which is our adenosine diphosphate, functions to increase platelet stickiness. And as more platelets stick, more platelets release these chemicals, and it snowballs. Would you call that a negative feedback response or a positive feedback response?
Positive.
Very good. Okay. And then thromboxane A2 does both of these. So thromboxanes are going to both increase vascular spasm and increase platelet stickiness. Okay. In fact, aspirin works on thromboxane. We talked about aspirin being described as a blood thinner. Okay, and aspirin prevents thromboxane synthesis. I actually had one of my students come up to see me after lecture this morning, and her sister had just had a baby last week by C-section, and her daughter ended up having her tonsils out, her niece. And so her niece was staying at her house since the mom was still in the hospital with the C-section, and she started hemorrhaging from the tonsillectomy. And so she had to have her admitted to the hospital, and the little girl actually had to have a transfusion. And the doctor said it was because the little girl had been given ibuprofen for pain, because the mother hadn't want to give her Tylenol. And my student was like, I didn't know ibuprofen. I thought it was aspirin that increased blood, was a blood thinner and decreased clotting ability. So we looked it up, and it said it had a very minor role. It only lasted about 2 hours in the blood, and so I said it shouldn't have caused her to hemorrhage. And she said, well, she has trouble healing anyway. and has a lot of bruises and stuff. And I said, well, when she gets, you might want to take her to a regular physician and have some blood tests done and see if there's some additional blood chemistry that's it, because ibuprofen shouldn't have caused a hemorrhage. I could see if it was aspirin or something, but normally it's very minor, and it shouldn't cause that much of a problem. So, anybody remember last time we saw thromboxane? Very first day of class. Thromboxane was in one of our groups of hormones at the very bottom. Under those derived from lipids. And steroid hormones were derived from cholesterol, and prostaglandins and thromboxanes were a group of eicosanoids derived from arachidonic acid. There you go. Right. We're doing a little bit of review for that comprehensive exam in 3 months, 2.5 months. All right, so these are found within that sac of plasma membrane pinched off from the megakaryocyte as the platelet. Okay? Now, platelet plug formation, again, is not a clot. Okay? It's just a bunch of sticky platelets. And we don't have to have any cells at all to have a clot. We usually will trap platelets, and we usually will trap red blood cells in our clot. And the sticky platelets can trigger a clot, all right? But we don't have to have platelets for a clot to occur. So here are the platelets getting sticky. And fibrinogen is one of our clotting proteins, and that whole process can be activated by the presence of platelets, but platelets are not required for that, all right? So down at the bottom, platelet aggregation, there are receptors for fibrinogen that can trigger a clot to form here. So that third mechanism is coagulation. Remember my favorite biological material?
Jello.
Jello. Okay, Jello is like a clot. So the fibrin proteins are the dissolved Jello proteins, and as they are polymerized as a result of our clotting reactions, they form long threads that interweave and trap the water molecules, and when the water can't flow, we have a clot. Okay? Also getting trapped in there would be the red blood cells and the platelets, but again, they don't have to. It's just the fibrin threads trapping the water is the clot. All right? So you have this diagram, or one on the next page, or a couple pages later, I have the steps actually written out because this is kind of a little fuzzy. But we have two pathways by which clots can be activated to form. Remember the dissolved protein threads, fibrinogen, is made by the liver, and it's found in your blood. And we walk a narrow line, or blood flows a narrow line, if you will, in which we don't want to have blood clotting all the time, right? But neither do we want to have the absence of clotting, which anytime you've heard of hemophiliacs, anytime you get a bruise, the blood doesn't clot and the vessels continue to seek blood. And so we have molecules in our body that prevent clots from forming, as well as these clotting factors. So, two pathways which can trigger a clot. One is when chemicals have to be added to the blood to trigger that. And that is known as the extrinsic pathway. The other pathway, no chemicals are added. It's just a different surface. It might be a glass surface. It might be the collagen fibers or an atherosclerotic plaque that trigger it. So no additional chemicals are added. It is triggered within the blood itself, and this is known as an intrinsic pathway. All right. So coming in contact with the platelets here, would this be an intrinsic or an extrinsic pathway?
Intrinsic.
Intrinsic, good, okay? So the extrinsic pathway requires damaged tissues. A crushing type of injury, not a clean cut through a vessel, all right? So my oldest son was telling me last week about what a hard week he'd had. He broke up again with his girlfriend, which he's broken up with her like 5 times in the last 2 years, and they've never actually gotten back together again. I know. She broke up with him 2 years ago in last August, and then she keeps telling him she's coming back, and then she doesn't. Does that make sense?
You should just
So,
leave her.
so yeah, well, he finally agreed last week that his mom and dad were right, but the next time she warms up to him, he's gonna be right there. Now, she broke up with him on his birthday, or the week before his birthday. She painted a 6-foot logo of the Kings logo on his bedroom wall, and then broke up with him. And then came back the next week with their parents to finish it. And then in February, he was a senior basketball team, and she comes to him and says, "If you win this game, I'll go out with you again." So they won, obviously with that kind of a carrot. And for 2 months, he kept trying to set a date. So finally, they had a date to go to movies with friends. And he talked to her the night before. I rearranged my schedule so he could have the car. And he calls her up like 2 hours before the movie and says, "So what time do you want me to pick you up?" And she says, "Well, I can't go. I have to clean my room."
I have to wash my hair.
Yeah. So every time she sees on Facebook that he's gone out with another girl, she sends him a little note, "How are you doing?" with a little smiley face. And he comes running back, and then as soon as he gets within pushing distance, she pushes him away. And anyway, so that kind of week. And he'd had a really bad sore throat, and he'd been out weightlifting and dropped a weight on his thumb, which is why I'm talking about this here. So it's big, swollen, and bruised. Okay? So I just had to get her out of my system. So this crushing injury where the damaged cells have released this chemical that has seeped into the blood, that's extrinsic. All right? A nice clean cut wouldn't be extrinsic. But this crushing kind of injury that's damaged the cells, and the chemical that's released is known as tissue thromboplastin. And that's the extrinsic factor. Okay, that's what's released from the cells outside of the blood vessel wall. Not to be confused with another molecule identified as a plasminogen, which gets converted to plasmin. That actually breaks down clots, okay? Thromboplastin triggers the clotting pathway. This breaks it down or prevents it from occurring. So tissue thromboplastin interacts with a clotting factor protein known as, and all of these are called factors, and they're given numbers. So you don't have to memorize any of these numbers except factor 10, okay? It requires calcium to become activated. And it interacts with factor 9. And none of these factors, unfortunately, are in any numerical order because they're numbered in the order in which they were discovered, not the order in which they work. I think they should go back and renumber them.
Yeah.
But anyway, they both come in to activate factor 10. Okay? 2 things here. Many of these factors are made by the liver. So guess what? When you have liver disease, hepatitis, liver cancer, cirrhosis of the liver, you're going to have difficulty with having your blood clot. You're going to bruise easily. Fatigue means not enough red blood cells. Easy bruising means not enough clotting factors or platelets. Okay? And what would be a sign and symptom of low white blood cells? Fever, getting infections, whatever. Okay. All right, or an infection would be present if you had a high number of white blood cells. But if you didn't have enough, you would get sick easily. All right, so calcium is required to activate many of these factors. In fact, if you have a tube of blood that you're working with and you don't want it to clot, often what is added is a substance, and there's a variety, known as a calcium chelator. And that's like a sponge. It binds to calcium and takes, it's kind of like calmodulin, binds to calcium and takes it out of solution so it can't interact with these clotting factors. And it prevents the blood from clotting. So it's one of the ways that we can store blood. Okay, so extrinsic pathway requires these cells to produce, these damaged cells to produce tissue thromboplastin. Intrinsic pathway could be a bunch of things. It can be the glass surface of a tube. It can be collagen fibers. It can be the inflamed lining of a blood vessel, phlebitis, which is an inflamed vein. Ever heard of DVTs? Deep vein thrombosis. Okay. A friend of ours was flying cross country, sitting the entire flight because he was in the middle of a wide-body jet, didn't get up and walk around. Came back, went to the gym to work out, and had an embolism, which is a moving clot. Fortunately, it didn't get to his heart. It was in his lungs, which can also cause death. He just had chest pain and went to the hospital, and they were able to give him clot-busting drugs.
Is it possible to go into coma if you have a blood clot in your lungs?
Usually a coma.
Or like something small, because we recently had a friend of ours who had a blood clot in his lungs, and he was like in a mini coma for.
You can certainly die from it if it affects a large region of oxygenation of the blood, and you're not getting enough oxygenated blood to the brain. It can cause a stroke if it's a clot that goes to the brain. A pulmonary embolism was the cause of the first death in the Iraq War. I think it was a journalist. So yeah, they can, you would think you have the other lung, why doesn't that work? But it can be fatal.
And can you repeat the function of the tissue thromboplastin and the?
So the tissue thromboplastin triggers the interaction between calcium and this first clotting factor and activates it. Otherwise, this clotting factor and calcium in the blood don't interact and don't trigger. This is another cascade or positive feedback, like a snowball that starts going. So the tissue thromboplastin interacting with calcium and factor 7 form a complex that activates factor 9, and both of those activate factor 10, and so on. So it's an overall kind of combination of that. All right, phlebitis, an atherosclerotic plaque, lipid material in the deep surface of the blood vessel. All right, any change in surface charge or regularity can trigger this intrinsic factor. So nothing new has to be added. Factors that are already present in the blood, factors 12 and 11, plus calcium activate factor 9. Again, you don't have to know those numbers because I can never keep them straight. And I try not to ask, well, I guess I do ask students to learn things I try not to ask students to learn things I can't keep straight. So, I can't keep straight my reasoning. So various factors, 12, 11, 9, combine together with calcium and also activate factor 10. The key thing that I want you to know is this does not require any additional chemical being added to the blood. So you can draw blood from the median cubital vein, put it in a glass tube with no new material added to it, and it will clot. Okay? Blood will clot in a blood vessel just standing there as clotting factors settle out and platelets get sticky. So we don't have to have damaged cells and torn vessels adding tissue thromboplastin. Once factor 10 is added, then we have a pathway that is in common. So everything follows this pathway from factor 10 on, and I want you to know that. It's this one right here. Okay, so factor 10 is going to activate an enzyme, again, already in the blood, made by the liver, known as prothrombinase. ACE indicates it's an enzyme. And this is going to cause prothrombin. It'll cleave off some amino acids and turn it into thrombin. And thrombin causes the fibrinogen to polymerize to those long threads I was talking about. And those trap water molecules, and we have a clot. Okay. So anything that affects the health of the liver So it can't make factors. Anything that affects the amount of calcium can affect whether or not, or affects prothrombin, will affect whether or not clots form. So here's an example of some of the factors. Even fibrinogen is given a factor name, and this is just for illustration. You don't have to memorize these, okay?
Professor,
Yes.
so just to make sure I got this straight, we should understand from factor 10 all the way to fibrinogen when a clot actually forms.
Right.
But nothing like the.
These factor numbers up here, you don't have to know that factor 7 is an extrinsic pathway and these factors are intrinsic. You just need to know that calcium is required for their activation and what triggers each pathway. So what's the triggering difference between intrinsic and extrinsic? And then from factor 10 down. So basically this and this. And the reason why we need calcium ions. Okay. So remember we talked about vitamin K when we were talking about circumcision? And vitamin K is made by the bacteria in the large intestine. So here's the purpose of vitamin K. The liver requires it as a coenzyme for making prothrombin. Okay, so prothrombin is made by the liver, and your body needs the vitamin K from those bacteria to be able to synthesize that. Now, what if we don't want a clot? All right, so some of these are natural factors and some of these are drugs that you can take. So platelets naturally repel each other like the same end of a magnet. They become sticky when they come in contact with the clot, the collagen fibers in the torn blood vessel, but normally they would repel each other, okay? Dilution of clotting factors. So all of those things that we talked about that increase the viscosity of the blood, dehydration and sweating, diuretics, would cause clotting factors to become more concentrated. So that's going to cause more clots to form. There was a situation, might have been more than one, but one that I read about a few years ago, where a woman had liposuction, and she scheduled some additional liposuction. It was going to be an in-office procedure, and her husband dropped her off in the morning and said, "I'll be back around 2 or 3 to pick you up." So we called about 2, and the office staff said, "Well, your wife's not ready yet." And he waited and waited, and he kept calling, and they kept putting him off and putting him off. Until finally about 5:30, he showed up and they said, "We're really sorry, but your wife has died." And what had happened is, I don't know how much it's progressed now, but the real basic mechanism of doing liposuction is a large cannula that's got a sharp edge, and they kind of slide it back and forth under the skin with a vacuum cleaner, not a vacuum cleaner, but a vacuum that sucks up the released lipid tissue and fluids. And of course they're cutting lipid tissue, adipose tissue has lots of blood vessels, so they're cutting blood vessels too. And in the hands of somebody that doesn't know where the major blood vessels are, they can do too much of an area and take up too much of the fluid. And so her blood pressure started to drop. So they started an IV, and instead of stopping the procedure, they kept going. And her blood pressure kept dropping, so they turned the IV up. And so what they ended up doing was gradually over that afternoon replacing her blood with intravenous fluid. Well, 2 things. Intravenous fluid doesn't have red blood cells and can't transport oxygen. So without oxygen, her tissues were starting to die. And when that happens, the capillaries in those tissues reach a point where they become like sieves. They just leak like crazy. And even if you add regular blood and oxygen back, you can't repair them. The other thing that you lose are clotting factors, okay? So again, if you add just regular blood back, there's not enough clotting factors to provide the clotting ability. You have to add concentrated clotting factors as if you were a hemophiliac to repair that. And there was a point, up to a point they could have saved her if they had admitted, okay, we're in over our heads and sent her to the hospital. But they weren't willing to do that until it was too late, and then even the best doctors couldn't provide enough fluid because everything just was leaking out of her blood vessels, and they couldn't stop that process. It gets to the point where it just is irreversible. So that dilution of clotting factors is usually a good thing. We don't want blood to clot, okay? But you can overdo it, certainly. And then anticoagulants. First, antithrombin 3 is something your body makes. The others are chemicals. Heparin is an intravenous drug that will activate antithrombin 3, prevents prothrombin from working on thrombin. Sometimes people need antibiotics twice a day by IV, but they don't need to be stuck to an IV pole 24 hours. So they'll do a needle into the vein called a heparin lock, and it has a little rubber diaphragm. And the IV is just injected slowly when they need the antibiotics, and then flushed with heparin to keep blood from backing up and clotting in the needle. And that way they can keep from re-injecting the person every time. Coumadin is an oral pill, all right, that interferes with vitamin K. So if someone has to be on an anticoagulant, my husband has a friend who, well, I guess he's my friend too, but my husband was a classmate, law classmate of my husband's, who was driving north on 99 in 89, just before Christmas, and they were working on the sound wall, and it was very foggy in December, and he looked in his rearview mirror and he saw this car coming on really, really fast. And he made a decision that he regrets to this day. He decided she was going to run into the back of him, and he decided to move over. And she did run into the back of him. And because he was moving over, she clipped him on one side instead of straight in the back. And that spun him around and sent him sideways into a Caltrans truck. And he broke his pelvis, and it just basically caved in the side of his car. The pelvis, because of the bone marrow and everything there, when that gets broken, it's a very fertile area for blood clots, for clots, thrombus, lipid material, coagulated blood. And so he ended up in the hospital for several weeks, and when he went home, I don't know, for some reason they never gave him a list of possible side effects, and he hadn't been home for more than a day before he started having chest pain. And his wife fortunately took him back to the emergency room, and he had sent a clot, an embolus to the lungs, and had a pulmonary embolism. And so to this day, that was over 20 years ago, he has to take daily doses of Coumadin to prevent blood coagulation. Red wine and red grape juice also have similar anticoagulant effects. My brother-in-law has lupus and takes Coumadin, and he has his blood clotting time. It's called pt.2, prothrombin time. And we used to do it in labs where we'd fill those capillary tubes up, and then after 2.5 minutes break it, and then 3 minutes, and then 3.5 minutes, and it would have finally a clot, stranded clot would form between the broken ends. And something that's an anticoagulant will extend that. It will have a long clotting time. So instead of your blood clotting between 2.5 and 3 minutes, it might be 3-5 minutes, which means a long PTT. And so he had his blood tested, and they called him up and they said, "How much alcohol are you drinking?" And he said, "I don't drink alcohol." And they said, "Well, you're drinking something." And I said, "Well, my father-in-law's here, and he drinks red grape juice for his heart, and I've been drinking that." And they said, "Stop. You can't do red grape juice and Coumadin at the same time." And because it was increasing his clotting time. Streptokinase, or TPA. TPA activates, it's a form of plasminogen that's created artificially in factories. And so it also, this is what's given to individuals that have a stroke due to a blood clot or maybe a heart attack, and it helps the clot break down. Aspirin inhibits the thromboxane A2, so it prevents the platelets from being sticky. Those are the primary ones. So in your lab, I ask for a list of clotting agents or anti-clotting agents, doesn't matter which or which ones, and to provide the mechanism of their activity. So any of these would be the anti-clotting agents. All right, so disorders of hemostasis, we've been talking about it. A clot is known as a thrombus. Okay, so it's a stationary clot in a vessel. If it happens to get loose, so let's say that you have phlebitis, or you've been sitting still on a plane for a long time with the edge of the seat compressing the back of your thighs and blood's not flowing well, a thrombus can form. If it's not moving, deep vein phlebitis often manifests with pain when you point your toes upward and stretch the muscles in your calf. So if someone comes in with pain in their calf and heat and swelling, you don't want to start rubbing their leg, okay? Because all that'll do is possibly break that clot off and send it off to the heart. Once it moves, it's known as an embolus or embolism. It doesn't have to be fat. You can have an air embolism. You can have a lipid embolism, okay? We're talking about blood today. And there's another clotting disorder that I wanted to mention. I used to have a case study called disseminated intravascular clotting disorder, or DIC. In the summer before I moved out to Davis to start my doctoral program, I had my RN for several years, and I was working at the hospital near my parents' house. I worked about 6 months in 3. So I was on their PRN staff, and I'd work 7 to 3, 3 to 11, come back at 7, work till 3, come back at 11, work till 3 the next day. Just pulling as much money as I could because I was paying out-of-state tuition. And my very last weekend, I was working labor and delivery, and we had a couple come in. They'd been trying for 10 years to get pregnant, and the day that their baby was due, the mom couldn't feel the baby move. Now, with my twins, I never felt them move. But she was concerned, and she went to the doctor's office, and they couldn't detect a fetal heartbeat. And so she sent them to the emergency room. The hospital went up to labor and delivery, and they confirmed that her baby had died. Their baby had died in utero. And so the parents and the doctor both wanted to do a C-section. And the other doctors at the hospital, they had privileges there, talked her out of it and said, you know, the physician, this is a major surgery, and you don't have any risk factors. The baby's life is not in danger, the mother's life is not in danger, and you'll be doing major surgery just to save their emotional stress. So they gave her prostaglandins, which spiked her temperature up to 105, and she was put on a cooling blanket that ice water was sent through. And after about 3.5 days, another nurse and I delivered her baby. There was nothing wrong with it externally. You know how when babies are born late, their skin sometimes sloughs off? This baby that didn't happen even though it died over 3 days before. It wasn't smelly. And the doctor arrived soon after. The other nurse and I delivered her little boy, and the doctor and the other nurse went in to deliver the placenta and all that, and I stayed with the dad. Cord wasn't wrapped around his neck. I don't know what the problem was. I left for California soon after, so I don't know if they even did an autopsy. I did go visit them before I left, and they decided they were going to adopt. They weren't even going to try this anymore. But the mother nearly died from bleeding to death because she had too many clots form in her body. She had disseminated intravascular clotting. So why would she die from having too many clots? She didn't die, but why couldn't she have died from having too many clots form? She was in surgery for 6 hours and had tons of transfused clotting factors in blood. So the couple of things, either the prostaglandins could have done it, or toxins from the tissue of the baby. This can happen with toxic shock syndrome. So women that wear tampons for too long cause a blood to clot, all right, and kind of an extrinsic factor, clotting mechanism, and tiny, not one big clot, but tiny, tiny clots throughout the body, using up all the clotting factors. So then, when her placenta was delivered, and the decidua separated from the stratum bassalis, there were not enough clotting factors left in her blood to stop the bleeding from that raw, nude surface of the uterus. And she nearly died from that. They had, like I said, they had to do multiple units of transfused blood and concentrated platelet factors to save her life. And I always found it interesting that she nearly died of bleeding to death because too many clots had formed, but it was the use. And I don't know, from case studies that students that did, several different causes came up, either the prostaglandins that were given to her to trigger the smooth muscle relaxation of the uterus, or toxins from the baby's tissue. But that can, like I said, that can happen with toxic shock syndrome, which we haven't heard about for a long time, but it's out there. This is an image of thrombocytopenia. One of our faculty here at AR has this. Thrombocytopenia is, this isn't her, but thrombocytopenia is insufficient numbers of platelets. And so again, we don't have the platelet plug forming. It's a different process than not having clots forming. The result is the same, easy bruising and bleeding into the tissues. All right, and of course hemophilia, which is a recessive disorder, often sex-linked, and not making, the most common is clotting factor 8. And we're not going to go into the recessive trait of it, just the fact of what it is. This is hemophilia. And then bleeding into the joints, similar to sickle cell effects. All right, now let's look at blood typing. So how many of you in here have had micro or had some other class where you talked about blood typing? That's all, 3 or 4, okay. Well, let me just, I'm going to pull the slide up, so let me just make sure I have gone through everything here. All right, so we're going to do this in lab, all right, and we're going to look at some samples of fake blood, unfortunately not your blood, and we're going to look for A and B and Rh factor or D antigens. When we find them, it will cause, the fluid that we add will cause agglutination. So agglutination shows the presence. This will be one of your questions on your lab quiz, all right? Agglutination shows the presence of the antigen. The red blood cells have been clumped together. If there is no clumping, agglutination hasn't occurred and the antigen is not present. All right, so we're going to go over this with you after we talk about blood typing and as we move into lab. But I wanted to give you a picture of what the actual agglutination looks like before I turn off the light. This table here you have in your lecture notes, okay? So that's kind of what I'm going to be diagramming on the board. Now in micro, you'll hear more about other blood types than just ABO and A positive, A negative, and I'll let them talk about that in micro. That's all I'm going to cover here. It's a little early. We haven't talked about antigens and antibodies yet, all right? But an antigen, by definition, and I'll give it to you again on Tuesday next week, is a substance or a molecule that triggers immune activity. It's circular reasoning. And immune activity is that activity triggered by an antigen. An antigen can be a glycolipid or a protein that has a variety of surfaces. And each one of these surfaces is recognized by that B cell or T cell receptor. Okay? And so the antigen is the whole thing, and it has different surfaces that are recognized more specifically. Normally, we do not have these antibodies against the antigen unless your body is exposed to it. So if you went and got a flu virus, a flu vaccination, that exposed your body to the various surfaces of the flu virus, and your body started making antibodies that match those surfaces, okay? But you didn't have those antibodies until you were exposed to it. In contrast, if you have A antigen on your blood, you are born with antibodies against the B antigen without ever being exposed to the B antigen by a transfusion, all right? So, as we make this table, blood type is identified by the presence of the antigen. So if you have the A antigen, you have blood type A. If you also have the RH antigen or D antigen, then we use a positive or a negative designation. So if you are A positive, you would have the A antigen and the RH factor or the D antigen. Okay? I'm going to set that RH factor aside for now and just look at the AB antigens first. So if your blood type is A, then the antigen present is going to be A. And you always have the antibody against the antigen that you don't have, all right? So the antibodies present and these are those Y-shaped structures that bind to cells, would be against the antigen B. Okay? Yes?
So are the antibodies and the
Agglutinins?
same thing?
Yeah, because they cause agglutination. Okay? Now, if you have the blood type B, that's because you would have antigen B on your blood cells, and therefore you would have the antibodies A roaming around in your plasma. Can you see a trend here? If your blood type is AB, you would have antigens A and antigens B. What antibodies would you have? None. Because if you did, you'd be agglutinating your own blood. Okay? Now, if you don't have any antigens, your blood type is O. Okay? Means no antigens. Since you have no antigens, you have antibodies against both. All right? So you would have A antigens antibodies and B antibodies. Okay, everybody with me on that? All right. Now, let's look at what blood types we can donate to. We don't want to get a transfusion reaction. So if I have a pool of blood here, all right, and I have A antigen, since I have A antigen, I'm going to have B antibody, right? Now what happens if I pour in B blood? All those B antigen blood cells will be agglutinated. Okay? And what I'm talking about is this image. Here is a red blood cell with B antigens. The antibody is specific to the B antigen and will bind to it, and it can bind to the same antigen on another blood cell. This is called agglutination. It doesn't itself, actually, the antibodies don't destroy the red blood cells. By agglutinating them, it makes them more likely to rupture. The antibodies don't cause that, but it makes them stick together so they don't go through blood vessels, smaller and smaller blood vessels very easily, and they're more likely to be engulfed by a macrophage. Okay? So I do not want to add any antigen to blood that has antibodies against it. Major rule. Okay. Now, so donate to. So A blood can donate to any blood type that doesn't have antibodies against A. I liken it to jumping in with the sharks. You never jump into a pool that has sharks against you. Okay? So I can donate A to A because it only has antibodies against B. So I can donate it to my same type of blood type. Can I donate A to B? No, because it has antibodies against A. Can I donate to AB?
Yes?
Yes, because there's no antibodies in AB against anything. So I can donate to AB. Can I donate to O? No, because O has both A and B antibodies. All right? Before I move on, does that make sense? So I cannot give blood to any blood that has antibodies against those antigens. All right, type B. Can I donate B to B? Yes. Can I donate B to A?
No?
Can I donate B to AB?
Yes.
Yes. Whoops, I meant to put just B up there. Can I donate B to O?
No?
No. Okay. AB. Can I donate AB to B? Nope, I've got antibodies against B. Can I donate AB to A? Nope, because I have antibodies against A. Obviously, I can donate AB to AB. Okay? Now, type O. Can I donate O to A? Yes. Now, O has antibodies against A. All right, so it's not the ideal solution. But the key thing is I can't add the antigen to the antibodies. It is possible in an emergency to add the antibodies to the antigen. But you can never add the antigen to the antibodies. If I add the antibodies to the antigen, they get diluted out, okay? So while I'm adding A antibodies to type A, there will be some agglutination, but I can still do it. If that's the only blood type I have and the person needs blood, okay? So I can add O to A. Wait, that's not the way I do. Cannot donate to O. I'm down here. So O can donate to A. O can donate to, am I doing this right here?
Yeah.
This is what I put in. Did this one wrong. AB cannot donate to, AB can only donate to AB. I did that one wrong. Okay, because I was donating to antibodies against. So O can donate to A, O can donate to B, O can donate to AB, and O can donate to itself. Okay? So, why can AB only donate to AB? Because I donate AB to A, I have antibodies against the B. If I donate AB to B, I have antibodies against the A. And if I donate to O, I have antibodies against both. Okay, in the blood that I'm donating to. So remember, what I can't do is I cannot donate to blood that has antibodies against the antigen being added. What is okay, so this is a no-no. I can't do that, okay?
Can you repeat all the rules again?
There's only one basic rule. You can't add an antigen to blood that has antibodies against it. Follow that rule, you'll be fine. Okay? What you can do is add antibodies to the antigen. That's okay. Not okay. But if you want just a simple rule, you can't add antigen to antibodies. So I can never